Oligosaccharides. Oligosaccharides contain 2 to 10 monosaccharides. Means they are made up of monosaccharide units, 2 to 10 monosaccharide units. On hydrolysis, oligosaccharides release monosaccharides. Hydrolysis is a process in which larger molecules break down into smaller molecules with addition of water. So on hydrolysis, oligosaccharides, larger molecules release, smaller molecules are their building units, that monosaccharides. On the basis of number of monosaccharides, they are divided as disaccharides, trisaccharides. On the basis of number of monosaccharides, means if they contain 2 monosaccharides, oligosaccharides contain 2 to 10 monosaccharides. So if they contain only 2 monosaccharides, they are called disaccharides. If they contain 3 monosaccharides, they are called trisaccharides. In this way, most commonly or most abundant oligosaccharides present in nature are disaccharides. Disaccharides, 2 monosaccharides held together by glycosidic bond. 2 monosaccharides combined together, their hydroxyl groups react with each other. Hydroxyl group of one monosaccharide react with the hydroxyl group of another monosaccharide to form a molecule or compound that is called disaccharide. Means 2 monosaccharides combine together to form disaccharide, and they combine through a bond that is formed between their hydroxyl groups, and that bond is called glycosidic bond. 2 types of disaccharides: reducing disaccharides, as disaccharides are also soluble in water and they are sweet. So disaccharide sugars are classified as reducing sugars and non-reducing sugars. Reducing disaccharides and non-reducing disaccharides. The examples of reducing disaccharides are maltose and lactose, and sucrose is non-reducing disaccharide. Reducing sugar or reducing disaccharide which contain free aldehyde or keto group. If any sugar contain free aldehyde group or keto group, it can be reducing sugar. But if any disaccharide or any sugar which does not contain free aldehyde or keto group, that is non-reducing sugar, because reducing property is due to presence of free aldehyde group or keto group. So sucrose does not contain free aldehyde or keto group. Maltose is composed of 2 alpha glucose units held together by 1,4 glycosidic bond. 1,4 means glycosidic bond means covalent bond formed between 2 hydroxyl groups. 1 and 4 means maltose is found composed of 2 alpha glucose units, means hydroxyl group of one glucose molecule at first carbon react with the hydroxyl group of another alpha glucose molecule at fourth carbon. So 1,4 glycosidic bond. Maltose is produced from hydrolysis of starch, and it is present in germinating seed. Starch is stored in the seeds, and when seed start to germinate, at that time metabolism starts and starch is converted into maltose. So maltose is present in germinating seed. Lactose is also called milk sugar because it is present in milk. It is composed of beta galactose and beta glucose by 1,4 glycosidic bond. Beta galactose and beta glucose by 1,4 glycosidic bond. Now sucrose. Sucrose is present in sugarcane and other sweet fruits. It is composed of alpha glucose and beta fructose by 1,2 glycosidic bond. Means hydroxyl group at first carbon react with the hydroxyl group at second carbon 1 2 glycosidic bond. Unlike maltose and lactose, in sucrose, reducing groups of both units, glucose and fructose, are involved in glycosidic bond. There is no free reducing group, so sucrose is non-reducing sugar. In maltose and lactose, these both are reducing disaccharides because they contain free reducing group. Because in maltose and lactose, their uh, reducing groups of their units, reducing groups of their monosaccharides, both reducing groups of their monosaccharides are not involved in glycosidic bond. One reducing group is present as free. So maltose and lactose are reducing sugar, but in sucrose, see, in maltose and lactose there is 1,4 glycosidic bond. In maltose and lactose there is 1,4 glycosidic bond. In 1,4 glycosidic bond, free aldehyde and ketone group of both their units. In lactose, galactose and glucose, and in maltose, both molecules of glucose. In maltose and lactose, there is 1,4 glycosidic bond. So in these disaccharides, the aldehyde and ketone groups of both monosaccharides are not involved in 1,4 glycosidic bond. One group is present as free, but in sucrose there is 1,2 glycosidic bond. So in 1,2 glycosidic bond, the both units, the aldehyde or keto group here, sucrose is composed of glucose and fructose. Glucose contain aldehyde group and fructose contain keto group. In 1,2 glycosidic bond, aldehyde group of glucose as well as keto group of fructose both are involved in 1,2 glycosidic bond. So there is no free aldehyde or keto group. Means functional groups of both units, glucose and fructose, are involved in 1,2 glycosidic bond in sucrose. So there is no free aldehyde or keto group. So sucrose is non-reducing sugar. Lactulose is a synthetic disaccharide. Lactulose is a synthetic. Maltose, lactose and sucrose, these are natural disaccharides, but lactulose is a synthetic disaccharide. It is neither digested in our body nor absorbed. असांजी बॉडी इनखे डाइजेस्ट भी न कंदी आहे, एब्जॉर्ब न कंदी आहे. इहो साइंथेटिक डाइसेक्राइड आहे, एंड इट इज़ यूज्ड इन हेपाटिक एनकेफालोपैथी. Hepatic encephalopathy. In hepatic encephalopathy, due to liver failure, ammonia builds in our body. Normally, liver metabolizes the ammonia, and ammonia does not elevate much more in the blood in our body. Ammonia is very toxic to the brain. When liver failure occurs and liver cannot metabolize ammonia, then concentration of ammonia in our body is increased, and it diffuses into the brain cells where it damages the neurons. It is very toxic for the brain. So in hepatic encephalopathy, lactulose is given orally. Lactulose is neither digested nor absorbed by our body. When it is taken orally in intestine, lactulose is converted into the lactic acid by the gut bacteria. And lactic acid, it is the acid due to acidic pH. Normally the ammonia which is synthesized in our gut, in our GIT, that diffuses from GIT lumen into the blood. But when lactulose is converted into the lactic acid in acidic pH, ammonia is a base, and in acidic pH ammonia accepts proton. This is ammonia. Ammonia is a base. Normally, this is non-polar and it easily diffuses from GIT lumen into the blood. But in acidic pH, it accepts proton and it become ammonium ion. So now this polarized charged particle ammonium ion cannot cross bowel wall, cannot go from lumen into the bloodstream. So in this way, concentration of ammonia in the body can be reduced by giving lactulose. So lactulose is used in hepatic encephalopathy. Now polysaccharides. Polysaccharides are large and complex carbohydrates. They are tasteless and not soluble in water. There are two types of polysaccharides, homopolysaccharides and heteropolysaccharides. Homopolysaccharides are those polysaccharides which contain or which are made up of a single type of monosaccharides. Means, for example, starch, inulin or glycogen. Starch is a homopolysaccharide because starch is composed of only glucose. No any other monosaccharide, only glucose. So our glycogen is also composed of only glucose, no any other monosaccharide. And inulin is composed of only fructose. It is a polymer of fructose. So homopolysaccharides are those which are composed of single type of monosaccharides. For example, only glucose or only fructose. And heteropolysaccharides, they contain glucose and galactose. They may contain glucose and galactose. So they are composed of different, or they contain different or a few monosaccharides. But homopolysaccharide contain only one monosaccharide. Example of heteropolysaccharide is mucopolysaccharides. Starch is a carbohydrate reserve of plant. Plants store their excess carbohydrate in the form of starch, and it is a dietary source of man and other animals. Starch is a dietary source of man and other animals. It is present in cereals, vegetables, roots, tubers. These all things which we eat. Through these things we take starch. There are two components of the starch. When we see the structure of the starch has two components in its structure: amylose and amylopectin. Amylose is a linear structure and amylopectin is a branched structure. Amylose is a linear structure. There is only 1,4-glycosidic bond. But in amylopectin is a branched, means there is also a linear structure which has 1,4-glycosidic bond, and at a branch there is 1,6-glycosidic bond also. Means at the point of branch 1,6-glycosidic bond is present. Inulin is polymer of fructose. It is water soluble because of its low molecular weight. It is used to assess kidney function by measuring GFR. Inulin is not utilized by our body, and it is used to measure the GFR. And measuring the GFR is assessment of kidney function. Glycogen is carbohydrate reserve of the animals. Animals are main. We store our energy, we store excess carbohydrate in the form of glycogen. Glycogen is also called animal starch. Glycogen is present in our body in liver and muscles. Its structure is similar to the amylopectin, means branched, means it has 1,4-glycosidic bond as well as 1,6-glycosidic bond.